Hello, my dear students. How are you today? I hope you are well. Before we proceed with discussing our lesson for today, I would like to say thank you to Sir Raymond Ledesma for allowing me to use this presentation. I just added some information that I think will be useful in our discussion. Before anything else, let us have our opening prayer. Let us bow our heads and feel the presence of our Lord. God of grace, we open our hearts, minds, and souls to worship to you. Thank you that today we dwell in your kingdom and live in your presence. Thank you that as we gather together, we join with all Christians across the world to glorify your holy name. Come be with us, inspire us, and lead us in our time together. We ask all this in the beautiful name of Jesus. Amen. For your attendance, just like the same with the previous pre-recorded lecture video that we have, kindly listen attentively and participate with the online virtual activities. You can use the comment section below in typing down your answer for every activity. Don't forget to include your complete name and section that will serve as your attendance for this class. Let us now proceed with your lesson for today. Our lesson for today is all about the genetic information and protein synthesis under module number 2, Perpetuation of Life. Here are the objectives for this lesson. Number 1 is to identify how the genetic information can be used to synthesize protein. Number 2 is to determine the amino acids provided with the tRNA or the transport ribonucleic acid. And the third one is to reflect with the complexity of life and identify the importance of protein. During our previous discussion about the reproduction of animals, we discussed the difference between sexual from asexual reproduction. That when we say sexual reproduction, it means that there is a certain union or the meeting of the gametes, the involvement of the gametes. While for asexual reproduction, it does not involve the union of gametes. We also know that reproduction or the animal reproduction is necessary for the survival of a species. For asexual reproduction, it produces a genetically identical organism, whereas in sexual reproduction, the genetic material of two individuals combines to produce offspring that are genetically different from their parents. We also discussed the importance of reproduction, wherein the main importance of this is to or is for the continuity of a certain species. We also discussed a type of artificial reproduction or fertilization, which is the in vitro fertilization. So when we say in vitro fertilization for our review, when we say IVF, it is a method of assisted reproduction in which a man's sperm and a woman's eggs or egg are combined outside of the body in a laboratory dish. One or more fertilized egg or the embryo may be transferred into the woman's uterus where they may implant in the uterine lining and develop. Okay, so for your first virtual activity for this lesson, you're just going to determine if the word or description below is for DNA or RNA. So if the description belongs to DNA, you're just going to write down or to type in DNA. And if it describes an RNA or a characteristic of RNA, you're just going to write down or to comment RNA. I am giving you 3 minutes to do this activity. You can use the comment section below to type in your answers, and then we're going to check it later. So you may start now. Before we proceed, let us have first a review or recap about the difference of DNA from RNA. So when we say DNA, short for deoxyribonucleic acid, the nucleic acid that is the genetic material determining the makeup of all living cells and viruses, resembling a ladder twisted into a spiral, or the so-called double helix structure. While RNA, or the ribonucleic acid, it is an important biological macromolecule that is present in all biological cells, and it is principally involved in the synthesis of proteins, carrying the messenger instructions from DNA, which itself contains the genetic instructions required for the development and maintenance of life. RNA and DNA are as follows. So, the first one is that RNA is single-stranded while DNA is double-stranded. Next, RNA contains uracil while DNA contains thymine. And then third one, RNA has the sugar ribose while DNA has the sugar deoxyribose. Okay, so let us now check your answers. So for number 1, the first one is single strand. So this description belongs to RNA. Next, number 2, double strand. This one is for, very good, the answer is DNA. How about number 3, with helical structure? So this one or this description belongs to DNA. And how about number 4, phosphate ribose backbone? When we say phosphate ribose backbone, So the sugar phosphate backbone forms the structural framework of the nucleic acids, including DNA and RNA. So the answer is both DNA and RNA. So that is correct. Next, number 5, with short strand. The answer is RNA. So why do you think? It is because DNA is longer than RNA. It is because DNA of cells holds all the genetic information of the organism. Next, number 6, uracil instead of thymine. What are uracil and thymine? Uracil and thymine are what? These are considered as some of the nitrogenous bases for DNA and RNA. So what do you think is the answer? For number 6, the answer is RNA. Very good. So in RNA, thymine is replaced by uracil as the base complementary to adenine. This base is very similar to thymine. So uracil is energetically less expensive to produce than thymine, which may account for its use in RNA. So that's why in RNA, uracil is used or produced instead of thymine. Number 7, ATCNG. What are these? ATCNG. ATCNG are the nitrogenous bases for DNA. So the answer for number 7 is DNA. Okay, so number 8. For number 8, sugar lacks 1 oxygen. So the answer for this item is DNA. So deoxyribonucleic acid. So from the term, it implies deoxy, which means containing less oxygen in the molecule. Next, number 9. It can be found in nucleus. The answer is very good. The answer is DNA. And the last one for number 10, it can be found in the cytoplasm. The answer is RNA. For our review, let us have first a review. Recall for this one. With regards in the DNA and RNA base pairing. So there is a certain rule to follow. For DNA to DNA, it includes the nitrogenous bases guanine, cytosine, adenine, and thymine. Guanine complements with cytosine, while adenine complements with thymine. And how about for DNA to mRNA? Guanine complements with cytosine, adenine complements with uracil, while thymine complements with adenine. And then last one, for mRNA, messenger RNA to tRNA, guanine complements with cytosine, while adenine complements with uracil. Okay, so let us now proceed with your second virtual activity. You're just going to write the complementary DNA strand for each given strand. Kindly take note of the DNA base pairing rule. So for cytosine, the complementary for the cytosine is guanine, and the complementary of adenine is thymine. So let us have number 1 or item number 1 as an example. So for number 1, we have the combination C, G, T, A, T, G, C, G, C, T, A, T, and A. Let us now write down or answer, or let us give the complementary strand for this. So for number 1, or for the first letter C, the complementary of C or the cytosine is guanine, G. And then G is to C, T is to A is to T, A is to T, T is to A, G is to C is to G, G is to C is to G, T is to A is to T, A is to T, T is to A, T is to A, and A is to T. Did you get it, my dear students? Nakuha ba, mga anak? Okay, so kindly do items number 2 and 3. So I am giving you 3 minutes to perform this activity, and then you can comment or type in your answers to the comment section below. Don't forget to include your complete name and your section that will serve as your attendance for this class. Okay, let us check your answers for items number 2 and number 3. For number 2, we have the strand or the combination TC, TTAAA, TGATC, GATNC. So the complementary for T is A, for C is G, T is A, T is A is for T, A is T, A is T, T is for A, G is for C, A is for T, T is for A, C is for G, G is for C, A complements with T, T is for A, and last one, C is for G. Did you get the right answer? Next, number 3. For number 3, we have the combination AA, TGAA, TAGC, TAGCTT. Let us now give the complementary strand. So for A, the complementary of adenine is thymine. So A is for T, A is to T, T is to A, G is to C, A is to T, A is to T, T is to A is to T, G is to C, C is to G, T is to A is to T, G is to C, C is to G, T is to A, and last one, T is to A. Okay, so let us now proceed with your next activity. So for your next activity, you're just going to write the mRNA strand for the given DNA strand. Kindly take note of the RNA base pairing rule, wherein guanine complements with cytosine, adenine complements with uracil, and thymine complements with adenine. Let us now proceed with item number 1 as your example for this virtual activity. So we have the combination for number 1, AT, GT, CG, CT, GA, TA, CT, and GT. Just take note of the rule wherein in RNA, thymine is replaced by uracil as the base complementary to adenine. This base is very similar to thymine, so uracil is energetically less expensive to produce than thymine, which may account for its use in RNA. So let us now deal with number 1 or item number 1. So the complementary of adenine is uracil. And then next, T or thymine is to adenine. Guanine is to cytosine. And then next, T is to A, C is to G, G is to C, C is to G, T is to A, G is to C, A is to U, T is to A is to U, C is to G, T is to A, G is to C, and then last one, T is to A. Kindly do items number 2 and number 3. You can comment down your answers to the comment section below together with your answer for the previous activity. Okay, so let us now check your answers. So for item number 2, the given DNA strand is GAAGCGATCAG So the answer or the mRNA strand complementary to the given DNA strand is for the first letter G. The complementary of G is C. Next is A is to U, A is to U, G is to C, C is to G, G is to C, A is to U, T is to A, C is to G, A is to U, G is to C, T is to A, T is to A is to U, C is to G, and last one, G is to C. Did you get the right answer? Next, item number 3. The given DNA strand for item number 3 is AA, TGAA, TAGC, TAGC, T, and D. So the answer, the complementary mRNA strand for the given DNA strand is For A, we have U. A is to U, A is to U, T is to A, G is to C, A is to U, A is to U, T is to A is to U, G is to C is to G, T is to A is to U, G is to C is to G, T is to A, and last one, T is to A. Okay, so let us now unfold. Let us try to differentiate the nucleic acids DNA and RNA. So DNA and RNA are considered as a long linear polymers called nucleic acids that carry information in a form that can be passed from one generation to the next. These macromolecules consist of a large number of linked nucleotides, each composed of a sugar, a phosphate, and a base. Now let us proceed with the primary function or with the function of these nucleic acids. So for DNA, the main function of DNA is for the repository of the genetic information. And how about RNA? The function of the RNA is to involve in the protein synthesis and gene regulation, carrier of the genetic information in some viruses. And how about the structure of these nucleic acids? So the structure of DNA is double helix, while RNA is usually single-stranded. Now let's talk about their bases. So the DNA is composed of the cytosine, thymine, adenine, and guanine. How about RNA? RNA is composed of the following nitrogenous bases, namely the cytosine, uracil, adenine, and guanine. So before we have explained why is it that uh, in RNA in RNA thymine is replaced with uracil. So our topic for today is all about protein synthesis. So let us first unlock the term protein. So when we say protein or proteins, these are organic compounds made up of amino acids and they function as any of the following. Proteins are macronutrients that is essential to the building muscle mass. It is commonly found in animal products, though it is also present in other sources, such as nuts and legumes. There are three macronutrients, so these are the proteins, fats, and carbohydrates for additional information, and macronutrients that provides calories or energy. The first function of protein in our body is that it serves as a structural protein. So when we say structural protein, uh, it help it helps the cells to keep their shape or make up muscle tissues. Second is that proteins act as a transport protein. Transport materials that transports materials in and out of the cell and around the body. An example of a protein that acts as a transport protein is the hemoglobin. A hemoglobin is a protein in your red blood cells that carries oxygen to your body's organs and tissues and transports carbon dioxide from your organs and tissues back to your lungs. If a hemoglobin test reveals that your hemoglobin level is low or lower than normal, it means that you have a low red blood cell count. or in other term, you are anemic or you have anemia. Aside from that, proteins also act as signals and receive signals. Next is that proteins serves as an enzyme which speed up a chemical reaction, or we can say a catalyst. And last one, proteins also act as or functions as an antibody. So an antibody, an example of a protein that serves as an antibody is yung tinatawag natin na immunoglobulin, which is a protective protein produced by the immune system in response to the presence of a foreign substance called an antigen. So antibodies recognize and latch onto antigens in order to remove them from the body. The process of protein synthesis. The process of protein synthesis started with the DNA. And this DNA should undergo two main processes, namely transcription and translation of DNA. When we say transcription, it is when the DNA is used as a template to make the messenger RNA, or also known as the RNA or the mRNA. And then later on, this mRNA will leave the nucleus and goes to the ribosome. And we all know that one of the main function of the ribosome is that it is being considered as the site of protein synthesis. Another process wherein DNA should undergo is the process of translation. Translation happens when genetic code in mRNA is read and used to make a protein. Ribosome reads the sequence of codons in mRNA, and molecules of tRNA brings amino acids to the ribosome in the correct sequence. tRNA, or the transfer ribonucleic acid molecule, has an anticodon for amino acid it carries. Anticodon is complementary to the codon on the messenger RNA. Okay, class, so for you to better understand the discussion for today, let us watch a short video clip about the topic protein synthesis. Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA. It is tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of the free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, A transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape, forming a protein. What we are going to present is just a glimpse of the whole process. Processes, including splicing and the use of enzymes to identify which part of the DNA should be moved or cut. So, we are just going to leave it to the higher form of sciences, but for the purpose of the basic acquisition of lesson about the central dogma, this is how are we going to deal with this. Okay, so let us now start. Let us analyze our illustration here in this slide. So as you can see here, for example, we have here for the DNA. Since DNA, we all know that DNA is composed of double strand. So this is the first strand. And then second strand natin. For the first strand, we have A, G, G, C, G, C, G, T, C, T, C, G, A, T, C. And then for the second strand of DNA, we have T, C, C, G, C, G, C, A, G, A, G, C, T, A, and G. And then the DNA, second strand of DNA, it will undergo the first process for it to move on with the process of protein synthesis, which is the process of, what do you call that process, class? That is transcription. So the strand of the DNA is being copied into a new molecule or the mRNA. So in talking about the second strand, it has a corresponding codon with or in becoming a new molecule. So for our second strand, we're going to consider this as the given. Okay, so for T, the complementary of thymine is adenine. Very good, that is correct. And for C, cytosine, the complementary of cytosine is guanine. Cytosine, guanine, guanine, cytosine, cytosine, guanine, guanine, cytosine, cytosine, guanine, and then how about for adenine? What will be the complementary of adenine? If the second strand of DNA is undergoing transcription, is it thymine? No. What will be the complementary of adenine if it is not thymine? So, the complementary of adenine is uracil. Since we are talking about the transcription, okay? And then, for guanine, the complementary of guanine is cytosine. A or adenine, uracil, very good. And then, guanine, cytosine, cytosine, guanine, thymine, adenine, very good. And then, adenine is to uracil. And then, last one, we have the guanine is to cytosine. Now, we have the complete strand for the mRNA. And then, it has a corresponding codon for the tRNA. We're just going to follow the given rule on how are we going to deal with giving the complementary for each nucleo or for each bases. Okay, so for adenine, the complementary of adenine is uracil. For guanine, the complementary of guanine is cytosine. And for guanine, again, cytosine. Cytosine is to guanine. Guanine is to cytosine. Cytosine is to guanine. Guanine is to cytosine. Uracil is to adenine. Cytosine is to guanine. Uracil is to adenine. Guanine or cytosine is to guanine or vice versa. And then guanine is to cytosine. Adenine is to uracil. Very good. And then uracil is to adenine. And then last one, cytosine is to guanine. And then in talking about the tRNA, we can unlock the, what do you call this one? The complementing amino acid by means of looking at the at these three bases that we have for the tRNA. So for example, we have the first set, which is U, C, and C. It complements the amino acid serine. And how do we know that UCC or uracil, cytosine, and cytosine is equivalent to a serine? So we will be using the so-called amino acid table. So for the amino acid table, as you can see here, we have here on the left portion, left column, we have here the first left column. Okay, the first nucleotide. So we have here the first nucleotide, which is U. So kindly take a look at the box for U or uracil. And then for the second nucleotide, ito yon sa taas. Second nucleotide is C. So from U, tingin dito sa C. Itatapat natin siya. We're going to match it with the third nucleotide, which is also C. U, C, and then C. 1, 2. Pangalawa. Nakakasunod ba? So then therefore, the combination of uracil, cytosine, and another cytosine is equivalent to the amino acid serine. Okay, next. For the second set, we have guanine, cytosine, and guanine. Now let us identify what type or what kind of amino acid will be formed if there is a combination of guanine, cytosine, and another guanine. So our first nucleotide is guanine. Okay, so guanine. And then followed by our second nucleotide, which is cytosine. So dito sa taas, C. Tatapatan natin sa portion ng guanine. And then our third nucleotide is guanine din. So tapat lang natin siya sa G. So then therefore, what will be the name of the amino acid? So the name of the amino acid with corresponding nucleotides G, CG is alanine or alanine. Did you get it? Nakakasunod ba mga anak? Okay. So let us proceed with the third one or third set. We have the combination CANG, CAG. First nucleotide C, second nucleotide A, and then third nucleotide is G, CA and G. So then therefore, the name of the amino acid is glutamine or glutamine. Next, fourth set, we have the combination A, G, and C. Okay, first nucleotide is A, second nucleotide is G, and then third nucleotide is C. A, G, and C. So then therefore, what will be the name of the amino acid for our for the fourth set? A, G, and C. A, G, and C. So the name of the amino acid is serine. Very good. And then last one, take a look at the last combination. We have the combination of the nucleotides U, A, and G. U, A, and G. Let us determine the name of the amino acid U, A, and G. Take a look at the amino acid table. U. First nucleotide is U or uracil, followed by A, adenine. And then, itapat lang natin siya sa G. G. So, what will be or what is the name of the amino acid? So, as you can see here, the name here is termination codon. What does it mean? That's why stop yung nakalagay. Stop, which means hinto in Tagalog. So, when we say termination codon or a stop codon, it is a codon nucleotide triplet within messenger RNA that signals the termination of the translation process of the current protein. So, ibig sabihin, kapag ka may stop codon na tayo dito sa dulo, it means na ano, natatapos na or huminto na yung translation process. Do you have any question? Okay, let us proceed with our next example para mas maintindihan pa. Next slide. Okay, class, so let us now proceed with your third virtual activity for this lesson. You're just going to determine the amino acids produced by decoding the strand of DNA below. Okay, so as you can see here on the slide, so we have the given for the DNA. We have cytosine, adenine, guanine, adenine, thymine, guanine, cytosine, guanine, adenine, and thymine. And then, on how are you going to determine the amino acid, you can use the given amino acid table below. So I'm giving you 3 minutes to perform this activity, and then kindly comment down your answers below, and then we're going to check it. Okay, class, let us now check your answers. Let us check if your answers are correct. So for the given DNA strand, we have CAGGAA, TTGC, TCGA, and T. So for the complementing mRNA, for the strand of mRNA, C is to G, A is to U, G is to C, G is to C, A is to U, A is to U, T is to A, T is to A, G is to C, C is to G, T is to A, C is to G, G is to C, A is to U. Very good, because we are talking about RNA, and then T is to A. And then from let us now give the complementing codon for the tRNA. So G is to C, U is to A, C is to G, C is to G, U is to A, U is to A is to U, A is to U, C is to G, G is to C, A is to U, G is to C, C is to G, U is to A, and the last one, A is to U. Now let us try to determine based on the given tRNA of what type or what kind of complementing amino acid is the combination of the given tRNA? So for the first set, we have C, A, and G, wherein it gives us the glutamine or glutamine. And then, second set, GAA, which is glutamate. And then third set, U, and G. We have leucine. Very good. And then fourth one, CU and C, leucine. And then last one, GA and U. So yung last combination natin is the amino acid aspartate. Okay, so I'm going to check your answers on the comment section. I'm going to like and to heart your comments if you get the correct answer. Okay, so we are done discussing our lesson for today. Let us now have the different points to remember. So the first one is that genetic material includes the DNA and the RNA, the deoxyribonucleic acid, and the ribonucleic acid. And then we all know that proteins are the biological molecules that have many functions in the living organism. So we have discussed a while ago that proteins exhibits different roles, such as being a structural protein. It helps in repairing wounds and cuts. And then aside from that, proteins also act as a transport protein. Sometimes they become a carrier of the material or the nutrients needed by the body. So an example of that is the hemoglobin. And then proteins also act as enzymes or catalysts, and sometimes they are also being considered as help, or they help with regards in dealing with the boosting of the immune system as an antibody. And then last one is the protein synthesis. Protein synthesis that includes the what? The different processes that DNA undergoes. So DNA undergoes the processes of transcription and then translation, and then having or producing protein. Aside from those things, you also learned today how to write the mRNA of the given DNA strand, and you also know how to determine the tRNA that will read the mRNA, and then you also know how to deal with the amino acid, or to identify the names of the amino acids by means of using the amino acid table. Okay, so let us now check your learnings for this lesson. So you're just going to answer this activity, multiple choices. You're just going to choose the letter of the best answer. So for question number one, what biological macromolecule has a function of repairing damaged muscles and serves as enzymes that speeds up the chemical reaction in the body, or that serves as a catalyst? Is it A, lipids, B, carbohydrates, C, nucleic acid, and letter C, protein? Now, what do you think is the answer? Okay, and then number two, kindly type in your answer first. We're going to check it after reading the entire questions. Number two, which nitrogenous base is not found in DNA? A, cytosine, B, guanine, C, uracil, or letter D, thymine? And then, question number three, it is the process in which cells makes protein. A, cell division, B, protein synthesis, C, genetic engineering, or letter D, reproduction? And then for item number 4, which of the following carries genetic code from DNA in a cell's nucleus to ribosomes? A, mRNA, B, tRNA, C, rRNA, or letter D, DNA? And then last question, number 5, what are the building blocks of protein? A, sugar, B, phosphate, C. Amino acid, or letter D. RNA? You can answer, you can start answering now. For your reflection, kindly pause and think about this. Number one is the complexity of life. Number two, the benefits of proteins. And then third one, technology keeps on improving. You can write down your reflection to our comment section below with a kindly use a minimum of 3 sentences. You can answer in English or in Tagalog. Thank you so much for attending my class today. And I hope that you have learned something. All the pictures and ideas used in the PowerPoint presentation are credits to the rightful owner.